Hello friends, we shall talk about the evolutionary significance of Prochloron, and the main objectives of today's deliberation are to give a brief introduction of Prochloron, to discuss in detail the fine structure of Prochloron, to study the evolution of Prochloron, to try and understand the pigment composition of Prochloron, and finally we shall try to learn the evolution of green algal chloroplasts from Prochloron. Let me first of all give you a brief introduction about the Prochloron. Prochloron, a prokaryotic alga, is an obligate symbiont of some tropical Ascidian colonies belonging to the family Didyminidae. In this symbiosis, the host Ascidian is thought to acquire photosynthetic metabolites provided from the symbionts, that is Prochloron, while the host provides a habitat for the symbionts. Radiolabeling studies suggested that Prochloron cells transfer photosynthetically fixed carbon to the host as soluble compounds of low molecular weight. Prochloron was discovered in 1975 by Ralph A. Levin of the Scripus Institution of Oceanography. Prochloron is one of the three known prochlorophytes, cyanobacteria, that contain both chlorophyll A and B bound to a special light harvesting protein. In the most host species, Prochloron cells are extracellularly distributed in the common cloacal cavity of the host, and the water currents there runs directly towards the outside of the host colony. Therefore, it is unlikely that the metabolites are effectively translocated to the host by means of diffusion, especially since there are no structures that are adapted for nutrient translocation in the cloacal cavity. The intracellular symbiosis of Prochloron cells is known only in Lysoclanum punctatum, but the host cells are free cells distributed in the integument of colony, that is tunic, and they do not have direct connection with the host zooids. Therefore, the occurrence of the metabolite transfer is questionable, since previous studies possibly underestimated the amount of the symbionts remaining in the host tissue when they determined the amount of labeled carbon supposedly incorporated into the host. In Diplosoma virens, the parasitic copypods that inhabit in the cloacal cavity are surrounded by numerous amounts of Prochloron cells. The copypods seem to feed on the tunic matrices but rarely ingest Prochloron cells. This may indicate that Prochloron cells contain toxic compounds to make themselves unsuitable as a food for the copypod. The host colony may be protected from predation by harboring the toxic symbionts, and this would be a considerable benefit to the Ascidians in the symbiotic system. Coming to our second objective, that is to discuss in detail the fine structure of Prochloron. The alga Prochloron is approximately spherical and 6 to 12 micrometer in diameter. Its fine structure is quite similar to that of a typical unicellular blue-green alga, except for a large central electron transparent zone. It appears to be identical to that illustrated for the prokaryotic symbiont of Diplosoma virens from the Great Barrier Reef and closely resembles that of Didymnum species from Baja California. However, the latter apparently contains a form of large storage granule not yet fully identified and not found in Diplosoma symbionts. The structure of Prochloron will be discussed under the following 4 headings, that is cell wall, thylakoids, cytoplasm and the electron transparent zone. First, we will discuss about the cell wall. As in other prokaryotic organisms, the cell wall of Prochloron is multilayered, although it appears to lack an external sheath. The wall is 35 to 50 nanometer thick, as in most blue-green algae. Zones of different electron densities equivalent to those found in blue-green algae can be distinguished with L first electron transparent, L second very electron dense, L third electron transparent, L fourth electron dense wavy. Internally, the cell membrane is always closely adpressed to the wall. Early stages of cross-wall formation in dividing algae have not so far been observed. However, a later stage of division, the cell membranes of 2 daughter cells are separated from each other by 2 L first zones, between which lies a very wide electron dense zone, probably L second. Only at the extremities of the cross wall, when separation of the daughter cells has already begun, can L third and L 4 zones be identified during cell division. Now coming to the second part, that is thylakoids. The thylakoids form a wide, usually peripheral band, but they are always separated from cell membrane and adjoining cell wall by a narrow zone of cytoplasm. An examination of sections of more than 100 organisms has failed to reveal any connection between the thylakoids and cell membrane. Phycobilisomes characteristic of blue-green or red algae have not been seen association with the thylakoids membranes of Prochloron. Unlike the single thylakoids of blue-green algae, those of Prochloron are stacked most often in pairs, but stacks of up to 12 have been observed. The tissue is unfortunately unsuitable for detailed analysis of the lamellar stacks. However, these stacked lamellae are not organized into grana, such as are typical of higher plant chloroplasts. Instead, they form discontinuous bands which resemble those of many green algae or the parallel lamellar bands of higher plant chloroplasts suffering from a deficiency of certain nutrients, for example, potassium. The occurrence in prokaryotic cells of stacked lamellae in conjunction with chlorophyll B as an accessory pigment is of particular importance in considering the origin of eukaryotic green algae and land plants. In the chloroplasts of light-grown higher plants, there often appear to be small, poorly defined centers usually associated with osmiophilic globules from which lamellae diverge. In Prochloron, similar small centers or junctions can also occasionally be distinguished. Coming to the third part, that is the cytoplasm. The cytoplasm appears to occupy two interconnected zones. The first forms a narrow band consistently 50 to 100 nanometer wide, lying between the cell membrane and the swollen outermost thylakoids. The second zone forms a matrix for the thylakoids and extends to the edge of the central electron transparent zone. The cytoplasm is of medium electron density and has associated with it granules of high electron density. The granules 15 to 20 nanometer often occur in lines or clusters and are similar in size to ribosomes. The cytoplasm also contains three types of inclusions. These closely resemble the osmiophilic globules, polyphosphate granules, and polyhedral bodies of blue-green algae. The polyphosphate-like granules are sometimes surrounded by electron transparent areas which lie within the ground cytoplasm and may contain occasional strands of fibrillar material. Structures such as fibrils, which could be interpreted as sites of DNA concentration, have not been observed within the general cytoplasm. Now, the fourth part is the electron transparent zone. It has not yet been possible to examine an entire prochloron by serial section. However, the approximate position within a cell of any section can be determined by the angle at which the cell wall is cut. All median sections of prochloron have a large central zone which is electron transparent and is therefore quite different from surrounding cytoplasm. This zone is absent from sections towards the edges of cells. Intermediate sections which graze the central zone show several small, apparently separate electron transparent areas. In dividing cells, the central zones are established before separation of the daughter cells is complete. The central zone of prochloron, from which the cytoplasm is excluded, is very different in appearance from the central nucleoplasm of most blue-green algae. The electron transparent has within it small, widely scattered groups of granules of varying size and electron density, occasional fine strands of medium electron density, and sometimes fragments or rings of membrane of greater electron density than the adjoining thylakoids. Structurally, the central zone is quite different from the surrounding granular cytoplasm. The two regions are separated from each other by membranous material. This is not in the form of a continuous membrane such as the tonoplast membrane of a higher plant cell. Coming to our third objective, that is to study about the evolution of prochloron. It is generally accepted that chloroplasts are originated from photosynthetic endosymbionts endocytized and retained by heterotrophic host cells, and that all chloroplasts share a common ancestor of cyanobacteria. In some algae, for example, red algae and green algae, and higher plants, the chloroplasts are thought to be directly originated from photosynthetic prokaryotes, that is, by primary symbiosis, and in the other algae, for example, brown algae, diatoms and dinoflagellates, the chloroplasts are originated from photosynthetic eukaryotes, where chloroplasts should have originated from prokaryotes through secondary symbiosis. Therefore, symbiosis of prokaryotic algae is of interest in relation to the evolution of chloroplasts. Prochloron, a prokaryotic alga, is an obligate symbiont of some tropical Ascidian colonies belonging to the family Didyminidae. This alga is originally described as a unicellular cyanophyte inhabiting the colony surface of Didymnum candidum and is characterized by containing chlorophyll A and chlorophyll B and lacking phycobilanes. Because this pigment composition is so unique among photosynthetic prokaryotes, the division Prochlorophyta was proposed for this alga Prochloron didymini. Since the pigment composition of prochloron is the same as that of the chloroplasts of green algae and higher plants, that is chlorophyta, prochloron may be closely related to the direct ancestor of the chloroplast in chlorophytes. Prochloron has been found in some other tropical didyminid ascidians, and the symbionts mostly inhabit inside the colony, that is, common cloacal cavity of the colony. Subsequently, two genera of free-living algae were subsequently found as the member of Prochlorophyta. First one is Prochlorothrix, that is a freshwater filamentous alga, and second one is Prochlorococcus, that is a tiny unicellular alga. The monophyly of the three genera is, however, not supported by molecular phylogeny. They are unlikely the specific ancestor of the chloroplasts, and each of them is a diverged member of the cyanobacteria. Therefore, the prochlorophytes are sometimes included in the division cyanobacteria. In contrast, phylogenetic analysis of chlorophyll B synthesis genes suggest that the genes of chlorophytes Prochloron, Prochlorothrix and Prochlorococcus share a common evolutionary origin. To date, Prochloron didymini is the only described species of the genus Prochloron. Prochloron cells are actually found in several didyminid species that are widely distributed in tropical waters, and thus Prochloron possibly consists of several species. There are, however, few differences in morphology among them. Moreover, there are no reliable reports on the in vitro culture of Prochloron cells, and thus there are no cultured strains even for the type species Prochloron didymini. This is the reason why this symbiotic alga is referred as Prochloron or Prochloron species in several reports. Although there have been some attempts to discriminate several groups of Prochloron based on cell size and cell structures, molecular phylogeny showed that Prochloron isolated from several host species and from geographically different sites are phylogenetically too close to be considered different species. Diversity of Prochloron is one of the unsolved problems that are essential to discuss its evolution and ecology. The biology of Prochloron has been studied from many aspects, for example, physiology, biochemistry, ultrastructure, and phylogeny. On the other hand, the host ascidians have been also studied for better understanding of this symbiosis system, but the actual condition of the symbiosis still remains to be disclosed. Now, we will discuss about the ascidian-prochloron symbiosis. The ascidian-prochloron symbiosis is undoubtedly a mutual symbiosis. Prochloron is an obligate symbiont of some digeminate ascidians inhabiting tropical waters. In rare cases, Prochloron is found on other invertebrates as epizoic film. Free-living Prochloron has never been found so far. On the other hand, the Prochloron-bearing ascidian species always contain the symbionts except for the host species in which the symbionts inhabit on the colony surface but not in the cloacal cavity, for example, Didyminum candidum. This means that Prochloron cannot survive without host ascidians, and Prochloron provides some benefits to the host to survive. Exceptionally, it is uncertain whether the epizoic Prochloron has specific association with the host. The host ascidian positively maintains the symbiosis. Vertical transmission of the symbionts usually occurs. The host larvae inherit Prochloron cells from their mother colony. The colony has the symbionts at the beginning of the life history, though the larvae need to have a tissue to catch and carry Prochloron cells. The occurrence of the vertical transmission suggests that the symbionts are important to the host for survival. Diversified adaptations for the vertical transmission are found in the host larva. Since the adaptations are different in each genus, there would be multiple origins of the symbiosis in the phylogeny of didyminid ascidians. For instance, the larva of Diplosoma species possesses specialized organs to collect Prochloron cell called plant rake or rostrum and to carry them. The embryo brooded in the tunic of the mother colony is free of Prochloron cells. A tassel-like structure protrudes from the posterior dorsal part of the trunk of the immature larva, and it is extended into the cloacal cavity to collect the Prochloron cells distributed there. Subsequently, the posterior half of the trunk extends posterior, forming a large pocket that packs the plant rake. That spawned larva carries Prochloron cell in the algal pouch, and then the algal pouch becomes the cloacal cavity in the metamorphosed larva or young colony. Now, we will discuss about the benefit obtained by Prochloron from this symbiosis. The cloacal cavity of the didyminid colony habitat for Prochloron is a highly protected space against predators. In didyminid ascidians, the cloacal cavity is usually sandwiched between the upper and lower layers of tunic, and the tunic contains acidic fluid and or calcareous spicules. Other organisms are rarely found in the cavity except for notodelphid copepods, indicating that Prochloron appears to be protected from competitors and predators. Ultraviolet radiation is a harmful environmental factor for survival, particularly in tropics, because of the severe irradiation and clear water there. In contrast, appropriate amount of photosynthetically active radiations is essential for photosynthetic organisms. Photosynthesis of isolated prochloron cells is affected by light intensity, pH, temperature and UV irradiation. In a prochloron bearing didyminid lysoclinum patella, the tunic contains UV absorbing substance identified as mycosporine like amino acids, and the prochloron cells isolated from the host are much more susceptible to UV radiations than those in the host. Prochloron cells are susceptible to UV radiations and they are protected in the host colony by a tunic wall transparent to PAR but not UV radiations. Ascidians also have benefits of this symbiotic association. In Ascidian prochloron symbiosis, the benefit of the host ascidians has been thought to be photosynthetic metabolites provided from the symbionts. Fisher and Trench in 1980 showed that prochloron cells release maximum of 7% of the fixed carbon mainly as glycolate in vitro. This indicates the symbionts have the potential to provide nutrient material to the host by secretion of soluble metabolite. There are some reports of radio-labeling studies showing that prochloron cells transfer photosynthetically fixed carbon to the host as soluble compounds of low molecular weight. In these studies, prochloron cells were isolated from the host after the incubation with carbon-14 for appropriate period in light or dark control, and then the radioactivities of the isolated symbionts and residual host colony were measured to estimate the amount of fixed carbon by photosynthesis and the amount of the transferred carbon to the host. Because it is almost impossible to separate prochloron cells completely from the ascidian colony, many prochloron cells still remain in the ascidian tissue after harvesting the prochloron cells. The ascidian host usually contains strong acid in the tunic. In intact colonies, the acid is stored in vacuoles of the tunic cells, but it is leaked during the isolation of the symbionts from the host colony. The acidic environment would denature the chlorophyll pigments and other molecules and may cause the miscalculation of the amount of prochloron cells and metabolites. In most host species, prochloron cells are extracellularly distributed in the common cloacal cavity of the host. The ascidians are usually filter feeders and they produce a continuous water current with the cilia of their branchial basket for filtration of food particles in seawater. It is unlikely that the photosynthetic metabolites are efficiently translocated to the host by means of diffusion. There are no structures that are adapted for nutrient uptake in the cloacal cavity. Although metabolite transfer is doubtful in the cloacal cavity, its possible existence cannot be ruled out. Furthermore, the histological study of the host colony demonstrated that the alimentary tract of the ascidian rarely contains prochloron cells, indicating that prochloron cells would not be usual diet of the host ascidian. Now we will briefly discuss about occurrence of the intracellular symbiosis. The intracellular distribution of prochloron cells was first reported in Lysoclinum volticicovae. Amoebocytes occasionally found in the cloacal cavity ingest the symbiont cells. Similar observations were reported in the cloacal cavity in Lysoclinum punctatum and the presumptive cloacal cavity of the larvae of Diplosoma similis. They are unlikely an occurrence of endosymbiosis because the process may involve subsequent digestion of the engulfed prochloron cells. The amoebocytes seem to spill from the ascidian tissue accidentally, and the amoebocytes laden with prochloron cells would probably be too large and immotile to migrate back to the ascidian tissue. Moreover, their relative frequency is too low, and thus the phagocytosis of prochloron cells in the cloacal cavity would provide few benefits to the host ascidians. The endosymbiosis of prochloron is demonstrated in the tunic of Lysoclinum punctatum, where prochloron cells are found not only in the peribranchial and cloacal cavities but also in the tunic and integumentary matrix covering the epidermis. Coming to our fourth objective, that is to study about the pigment composition of prochloron and its significance in the evolution of green algae. All green algae contain chloroplasts with photosynthetic pigments chlorophyll A, chlorophyll B and carotenoids. Cyanobacteria, on the other hand, only have chlorophyll A and phycobilins, while as phycobilins are absent in green algae and land plants. Prochloron is closely related to cyanobacteria but differs from other cyanobacteria because it contains both chlorophyll A or B and lacks phycobiliproteins. There are three known prochlorophytes, that is Prochloron, Prochlorothrix and Prochlorococcus. No other prokaryotic life forms contain both chlorophyll A and B, but all photosynthetic eukaryotes, land plants and green algae contain chlorophyll A and B. At present, there are studies being carried out on the photosynthetic machinery of prochloron because its morphology is distinctive among prokaryotes. Coming to our last objective, that is to study about the evolution of green algal chloroplasts from prochloron in terms of endosymbiotic theory. Evolutionary pathways for the origin of plastids in support with the endosymbiotic hypothesis is one of the most interesting and complex problems in the field of evolution and development. It was a great finding to discover a bacterium like prochloron which contains all the light harvesting pigments which are present in chloroplasts. All green algae have chlorophyll B, so the common ancestor of all green algae must be having chlorophyll B. According to the endosymbiotic theory, this common ancestor should have been the host of the previously free-living symbiotic bacterium, which ultimately developed into a chloroplast. Hence, the green algae must have acquired their plastids from the endosymbiosis of a photosynthetic bacteria having chlorophyll B, such as Prochloron. It is believed that the outer membrane of the chloroplast envelope represents the vacuolar membrane of the host cell, and the inner membrane represents the cell membrane of the photosynthetic bacteria. The unique photosynthetic machinery of Prochloron among cyanobacteria makes them fascinating to scientists who investigate the evolution of the characters in photosynthetic organisms. Learning more about the genetics of Prochloron will help researchers to determine relationships between these bacteria and other photosynthesizers, especially because of their distinct functional and structural photosynthetic features. Prochloron cells are structurally comparable to the photosynthetic machinery of green plastids, having the thylakoids placed in the cytoplasm of the bacterial cells. This feature forms the basis for the assumption that Prochloron is similar to the symbiotic bacteria which evolved into the first chloroplast within the autotrophic eukaryotes. The light harvesting pigment composition of Prochloron has similarity with both cyanobacteria and chlorophytes. In terms of endosymbiotic theory, it was proposed that Prochloron or its ancestor is the most probable precursor of the chloroplast found in photosynthesizing eukaryotes, and their chlorophyll B containing chloroplasts too can be assumed to have arisen originally by the symbiosis of Prochloron-like prokaryotic cells. This brings us to the end of the lecture on evolutionary significance of Prochloron. Hope you have understood it well. Thank you and goodbye.